The topic for the present video is specification for pharmaceutical formulations. I thought to make this video because every pharma professional should know about the specifications. So, specifications are the list of requirement for the test and the limits of those particular tests which are required to be fulfilled or which are required to be pass or which are required to have certain criteria to demonstrate the quality of the pharmaceutical formulations. The specifications are required and are must for ensuring the quality, safety and efficacy of the formulations or the drug products. The specifications are not only for the finished products, but these are also for the raw materials like drug substances, excipients, in-process stage materials, and finished formulations. Also, specifications are there for packaging materials. Pharmaceutical specifications define the criteria a drug product must meet to ensure quality, safety, and efficacy. These specifications are vital for regulatory compliance, product consistency, and patient safety. If the formulation or raw material or packaging material doesn't meet those specifications for quality, safety, and purity, then these are required to be retested and rejected. So, some criteria are there for specific test to fulfill by the material to demonstrate the quality. Specifications or specification is a list of tests, analytical procedures, and acceptance criteria. Acceptance criteria are the numerical limits or ranges that define the quality of the product. For example, the specification of assay. The assay limit should come within certain limit or range, that is 90% to 110% for some markets and 95 to 105% for some markets. So assay is a part of specification, and assay by HPLC or UV is a analytical procedure or analytical process, or you can say the analytical method which is denoted, and the acceptance criteria, that is specification, is 90 to 110% or 95 to 110%, 95 to 105%. Specifications are required to establish the quality standard for drug substance, drug product, packaging material, and raw materials. They are critical quality standards that are proposed by the manufacturer and approved by the regulatory authorities. So specifications are based onto the product behavior, product performance, and it is required to be approved by the regulatory authorities. Guidelines are required to be studied, and guidelines and the pharmacopoeial monographs are required to be studied for making the specifications. Specification justification is required to get the regulatory approval. Specifications include information about the substance purity, identity, potency, and other parameters. They also include the test methods that will be used to measure these parameters. Specifications are important because they have a significant impact on quality of the medicines. Specifications are presented in detail of the certificate of analysis, that is COA of raw material or finished product or in-process material. So you might have heard about the word COA, that is certificate of analysis. So all these tests, their procedures, criteria, and whether the product or raw material complies or doesn't complies is given in the COA. So key components of the pharmaceutical specifications are like description, strength or potency, purity, content uniformity, dissolution or disintegration, microbial quality, moisture content, and pH. So these are some examples I have given. The specifications are specific for the product, specific for the strength, dosage form, and these are specific for the different formulation designs. Description is the product's appearance. Identification confirms the drug is what and is supposed to be. For example, identification is done by UV and HPLC to identify whether the product supports the label claim or not. For example, you are making a tablet of aspirin. So first identification will be there. Identification test will confirm whether the given powder is of aspirin or not. Or identification of the aspirin tablet will give you information that the tablet is of aspirin or not. Strength or potency to ensure the correct API content. Purity, check for the impurities and contaminants. Content uniformity, ensure consistent API doses in each unit. Dissolution, disintegration to evaluate the release behavior. Microbial quality, ensure product sterility and bioburden limits. Moisture content, control for the stability and for the flow properties. So moisture content or water content is there. Sometimes LOD testing is there. pH is to ensure appropriate pH for stability and bioavailability. These are some of the components of specification. I have taken this information only to make you understand about the specification tests. Always remember that the specifications differ by product to product. Maybe less test for some product and higher number of tests for some other products. The tests are required to demonstrate the quality, safety, and purity of the formulation. For example, I have taken some tests, identify identity of the pharmaceutical product. The purpose is to confirm the drug is identified. Chemical identification by UV, IR, HPLC, and physical appearance by color, size, and shape. So description and identification. Strength or potency to ensure correct amount of API is present. Assay of active ingredient is the example of strength and potency. Techniques like HPLC, UV, spectrophotometry, and titration can be used to denote the strength and potency. Purity of the pharmaceutical product. Purity is confirmed by impurity testing, whether the impurities are within acceptable ranges or not. Impurity profiling is done by HPLC, GC. So impurities may be organic, inorganic, metal impurities, residual solvents, and many other. Nowadays, in the pharmaceutical field, nitrosamine is also being asked as a impurity content. Limit test for specific contaminants like heavy metals, residual solvents, etc. for purity testing. Content uniformity to ensure dosage units contain the correct amount of API. Generally, the formulations like tablets and capsules are tested mainly for the content uniformity. Also, the multi-dose formulations are tested. Individual doses unit assay is done in the content uniformity testing. Dissolution and disintegration testing to evaluate release profile and bioavailability of the drug. Dissolution test to measure the API release in the simulated GI environments or not, or other environments as required by the formulation. Disintegration test check if the solid doses form break down appropriately. Also, these testings are done onto the initial release and stability samples. Microbial quality, that is sterility and bioburden, to ensure the formulation is free from harmful microorganisms. Sterility testing for injectable and sterile formulations. Bioburden testing for non-sterile formulations. Endotoxin testing for injectable formulations. Moisture content, water content and LOD to ensure the levels are controlled for the product stability. More water content may lead to hydrolysis, may lead to change in the description, may also lead to generation of the impurities. Loss on drying measured by weight loss, and water by KF is measured by Karl-Fischer titration to provide precise moisture content determination. pH of pharmaceutical formulation. Measure the formulation has the right pH for stability and efficacy. pH measurement determines the pH of liquid, suspension or gel formulations. Particle size distribution for suspension, powders, creams. Control formulation characteristics like flow, stability and release rate. Laser diffraction or microscopy measures particle size distribution. So these are the product specific test. Different product have different test and the release criteria or specification criteria, or you can say the acceptance criteria. Viscosity of liquid formulations to ensure appropriate flow properties of manufacturing and use. Viscosity measurement is done by viscometer or rheometer. Stability of pharmaceutical formulations. Ensure product maintains efficacy and safety over time. Stability studies conducted under accelerated and real-time conditions in different packs. Retest and expiry date defined through the stability testing and real-time testing. Based on the data, the retest and expiry date is given. Excipient quality and compatibility to ensure the excipients are of high quality and do not interact adversely with the API. Compatibility studies are also done, and these are done to ensure excipients don't affect the API stability and efficacy. Coming to the types of pharmaceutical specification. So pharmacopoeial specifications are there. You can remember it like by the monographs or pharmacopoeial monograph. Based on that, pharmacopoeial specifications are there, and these are standard from the official pharmacopoeias like USP, BP. Then in-house specifications. These are the manufacturer specific standards for product consistency. Then regulatory specifications. Requirements from regulatory agencies like FDA, EMA or other authorities. Then another classification can be in-process specification, release specification and the stability specification. So in-process specification is to release the material after testing for the next process step, and release testing is for releasing the product into the market or sending it to the other client. And stability specifications are for checking the stability of the formulation and the APIs. Coming to why specifications matter, because specifications are important for ensuring the product quality and safety and efficacy. Specifications ensure product meet the safety, efficacy and quality standards. Specifications are very much important for regulatory compliance, critical for approval and market access, patient safety, prevents harmful impurities and ensures proper dosing. So whenever specifications are there and these are meeting, then batch can be released for the public. Consistency guarantees uniform product and uniformity across the batches. So these are the important parameters for specifications. These are required for ensuring the quality, safety and efficacy. Specifications cover various aspects like identity, potency, purity, dissolution and stability. all of which are critical to product development and regulatory approval. Whenever there are requirements of change in the specifications, then a lot of data is required to be presented and generated so that the specification can be justified. By adhering to specifications, manufacturers ensure consistency, safety and compliance in the pharmaceutical products. If the formulation does not meet the specifications, then the product is said to be failed, or there is a out of specification, that is OS may be there, OOT may be there, that is out of trend, and there may be out of expectation results. That's why critical specifications should be met, acceptance criteria should be met, specifications are required to be designed properly. For making the specifications, ICH guidelines are there. Regulatory body specific guidelines are also there from USFDA, EMA, ANVISA, and many other regulatory bodies. So all are required to be understood for meaningful specifications. Specifications should be as per the regulatory requirements. So this was the information for specifications for pharmaceutical formulations. I hope you might have got good information, and if you have any queries, please comment. Thank you for watching the video, and please share these videos to your colleagues and friends. Thank you.